and thank you for being part of this taped interview, which is part of Bionews Services' official coverage of ECTRIMS 2017. Gilenya, that is fingolimod, is approved to treat relapsing MS in adults, while no disease-modifying treatment currently exists for children and adolescents with MS. And the ongoing Paradigms phase 3 trial is testing Gilenya in children and adolescents. Data from that trial suggests that oral Gilenya can decrease relapses in pediatric patients aged 10 to 17 when compared to Avonex, which is interferon beta 1A, over a period of up to 2 years. How clinically meaningful is this decrease in relapses for children, would you say?
First of all, thanks. It's a great question because we are much less used to read the data from pediatric MS. I would suggest to take a little bit step backwards and understand a little bit more about pediatric MS. Approximately 3 to 5% of the 2.3 million MSers in the world are pediatric MS, ages 10 to 18. And there are a couple of important things that we need to know about this disease. In general, pediatric MS is much more active in terms of relapse rate and in terms of accumulation of MRI active lesions as compared to adult MS. What does it mean for the patient? For these patients, a relapse, which is actually a manifestation, any neurological manifestation, be it cognitive decline, vertigo, numbness, or problems with walking, is much more dramatic and severe as compared to adult MS. These manifestations are much more frequent. And if you think about a 12 or 13 or 14-year-old girl, which starts to understand where she is, and body image, mood swings, and things which are, I would say, very common in this patient, in this population in general. When you add to that such severe relapses that are treated by boluses of high-dose corticosteroids, which are treated with high doses of corticosteroids, this is a major impact on quality of life. These patients need to go to school, need to be with their friends, to play sports, and these very common and debilitating relapses make their life quite miserable, I would say. And as you rightfully said, there is no specifically indicated therapy for pediatric MS. And this paradigm study is I would say, first ever conducted, randomized, double-blind, controlled study in this patient population. So, in terms of relapse reduction, we saw 82% reduction in favor of fingolimod as compared to interferon beta 1A injections, which are pretty much one of the non-validated but standard of care therapies in pediatric MS. 82% reduction is something in terms of the magnitude of the effect, which was of course highly statistically significant, is something that we have not seen in other MS studies. Usually we are used to effect sizes of between 30 to 50, 60%. 82% is something which we consider as highly significant in this patient population.
And how would you say that the treatment impacted these children's disability levels and quality of life?
So that's another great question. So we know in general that these patients' relapses, as I've said before, are much more severe and much more dramatic. And this leads to accumulation of disability. They actually convert to SPMS, to secondary progressive MS, approximately 10 years earlier as compared to adult patients. So we did assess confirmed disability progression in this study as an additional analysis. The results, which will be communicated at the session, I can tell you that qualitatively they were very positive. There was a very good reduction, very significant reduction of confirmed disability progression, 3 months confirmed disability progression in fingolimod treated pediatric patients as compared to those who were treated with interferon beta 1A.
And did you gather quality of life data as well?
Quality of life data was gathered, and they will be communicated. We are actually analyzing the data right now. They will be communicated in further publications, for sure.
So in adults, Gilenya has been shown to improve four measures, important measures of relapsing MS, which are relapse rates, MRI lesions, brain shrinkage, and disability progressions. So were these four measures also analyzed? You spoke of relapse rates and disability progression. How did MRIs look in this population? And did you analyze brain shrinkage?
Great question. So for sure you're absolutely right. For Gilenya in adults, these four measures of disease activity are a clear differentiator of the high efficacy that this drug has in terms of its very novel mechanism of action. So with a lot of caution, because we know less about pediatric MS, let's be humble, and we do know that the entire pathophysiology is most probably similar to adult MS. We know that they accumulate, as I've said before, more lesions. They are much more active. And I can tell you that there were voices that told us that given the very high activity of these patients, we may not be able to show efficacy in this patient population because they are very, very active. So we did what we could. For example, brain volume loss is in adults is associated with cognitive decline and physical disability. We know, as I've said before, much less in the pediatric population. We did test everything that we are used to in adult MS in pediatric MS. So in terms of relapses, as I've said before, 82% reduction in relapses, which appears to be quite significant. In terms of new T2 or gadolinium-enhancing T1 lesions, the active MRI lesions, again, positive results which are confirming the robustness of this efficacious drug in terms of focal disease activity. The actual numbers will be presented at the podium presentation on the 28th. In terms of confirmed disability progression, the same 3 months confirmed disability progression. We showed fingolimod managed to show a beneficial effect as compared to interferon beta 1a in this additional analysis. And in terms of brain volume loss, a couple of words on brain volume loss. We are used to brain volume loss to brain shrinkage in adults on the background of an already shrinking brain. You know, it's unpleasant to know, but all of us here, our brains are shrinking. In kids, their brains are growing. So it was highly complex to make this analysis, but on top of despite this complexity, I can tell you again, the numbers will be presented at the platform presentation. Despite this complexity, fingolimod managed to show reduction, or actually slowing down, brain shrinkage in pediatric patients with MS in a statistically significant manner. So we're waiting forward to present this data in detail.
Thank you. So let's speak a little bit about side effects of Gilenya. In adults, you can see dizziness or headaches, nausea, cough, fatigue. Were children experiencing the same types of side effects? And also, in adults, adults are, as I understand it, monitored for changes in heart rate. Were children also monitored for heart rate changes?
Correct. So again, maybe 2 centimeters backwards, for sure. And I think that you know that from our experience with Gilenya in adults, the patient safety is of utmost importance to us. And particularly in the pediatric population, they are much more vulnerable to everything. In general, the safety profile that we know from clinical trials in adults with fingolimod, with Gilenya, let me put it this way. The safety profile that we saw in the Paradigm study in pediatric patients was in line with the safety profile that we know, that we are well acquainted with in adults, Gilenya in adults in clinical trials. Now, you mentioned a couple of adverse events. We need to, the exact data will be shown in the platform presentation. Overall, I can tell you that the incidence of adverse events was even a little bit higher in the interferon beta-1a as compared to fingolimod. We need to put it into the context because we are used to analyze safety data in 30, 40, 50, and maybe some older patients, and here we're analyzing safety data in kids and adolescents. So this needs to be put in the context of the age, and there are certain adverse events that are more common in general in an older patient population. But as I've said before, we are pretty much reassured about the safety profile that we saw in the Paradigm study. You asked about heart rate. Yes, we know very well the well-characterized pharmacodynamic effects of fingolimod upon treatment initiation in terms of heart rate lowering effects, and in common practice, these patients, adult patients, are used to be monitored for 6 hours upon treatment initiation. We did the same in the pediatric population. They were monitored just the same, and data was very reassuring and will be presented as well at ECTRIMS. You mentioned also PML. So PML with Gilenya in general is very rare in the adult population. To start from the end, we asked physicians to be extremely vigilant to every sign, clinical or imaging, of infection in general. These patients, these pediatric patients in Paradigms, had very frequent visits, underwent neurological examinations, and frequent MRI imaging. There was no PML reported in this study. Full stop. However, we studied, out of the 215 patients in this study, 100 of them were on fingolimod for up to 24 months. It's not a lot. We are, as Novartis, we are never satisfied in terms of our eagerness to have more knowledge. So there will be actually ongoing open label extension for 5 years for these patients. So we'll gather more safety information.
So the trial appears quite successful. Is Novartis planning to submit a request for approval in Europe and in the United States for a pediatric population?
We are extremely happy about the success of the study. We believe that it can be a life-changing for pediatric patients. And yes, we have started discussions with the regulatory authorities, FDA and EMA, in order to bring this drug to pediatric patients as soon as we can.
Thank you. So let's speak about another study, the EXPAND study testing siponimod. It presented data showing quite a robust treatment effect in terms of slowed disability progression, also reduced lesion burden and slow brain wasting in secondary progressive MS patients. So the trial is planned to run until 2023. It began in 2012. Is Novartis planning to submit regulatory filings for siponimod as an SPMS treatment based on current data?
So, as you said, the trial, the core phase of the study read out in the summer of 2016, showing these robust effects: 21% reduction in 3 months confirmed disability progression, 26% reduction in 6 months confirmed disability progression, both highly statistically significant in a placebo-controlled study, actually the largest study ever conducted in SPMS. SPMS is a huge unmet need. There were a couple of recent failures in this patient population in terms of clinical research, and we know that most RRMS patients convert eventually to SPMS. The timeline that you mentioned, 2023, is actually the readout of the open label extension of this study, because all of these patients continue to be followed up from efficacy perspective and safety perspective, because we want to have more data. We do believe that this drug can change lives of people with advanced disease, with SPMS, and we plan to submit. We have a couple of good regulatory interactions with FDA. We plan to submit for SPMS in the first half of 2018 in the US, and in the second half of 2018 in Europe. The exact wording of the label will be, of course, a matter of discussion and negotiations with the regulator, but we expect that this unique patient population and the unique results obtained in SPMS in this study, in the EXPAND study, will be reflected in the labels.
Investigators for the EXPAND study testing siponimod presented data showing robust treatment effects in terms of slow disability progression, reduced lesion burden, and slower brain atrophy in secondary progressive MS. In this trial, which goes into an open extension phase until 2023, is Novartis planning to submit regulatory findings for this treatment as in secondary progressive treatment based on the data that has been collected and reported to date, or will it wait for the open extension? And if so, do you know when you might be filing?
So yes, we are very happy with the data that was first presented in 2016, and with additional efficacy data that was presented at ECTRIMS. It was the first study that showed robust effects on confirmed disability progression, either 3 months confirmed disability progression or 6 months confirmed disability progression, in the ranges of 21 to 26%, highly statistically significant, and as you rightfully said, accompanied by evidence of efficacy on T2 lesion counts and brain volume loss in this unique patient population with SPMS. The answer to your question is yes. We are planning to submit in SPMS. We had some fruitful discussions with FDA. We're planning to submit in SPMS the first half of 2018. The exact label wording will be, of course, a matter of negotiation and discussion with the regulatory authorities. We need to remember that SPMS, as I've said before, is a continuum of RMS, so sometimes it's difficult to dissect between RMS and SPMS. But regardless, we expect that this unique effect and the unique patient population studied in EXPAND will be reflected in the label. In Europe, we're planning to submit in the second half of 2018 in SPMS.
Excellent. That is good news for this population. Can you briefly tell us then exactly how this treatment slows progression in these patients?
So first of all, we need to take a step backwards and understand what SPMS means. SPMS is a continuum of relapsing remitting MS. Unlike in RRMS, where the level of disability between relapses is constant, in SPMS, in between relapses, which become increasingly rare over time, the level of disability is increasing. The central nervous system gets into a sort of an autopilot, autonomous mode of independent disability progression. Independent means independent of the focal damage of the relapses. So this is something which poses a huge unmet need for patients, and as far as I recall, there is no therapy that was proven to be efficacious in this patient population. In terms of the pathophysiology, it is translated to accumulation of focal damage, which is very similar to the damage that we see in the classical relapsing remitting MS, but much more pronounced diffuse damage that is characterized by axonal loss, demyelination all over the brain, including brain shrinkage and so on and so forth, cognitive decline, and many other deleterious consequences for the patient. How does siponimod impact this thing? Siponimod is a small molecule second generation novel S1P1 and 5 receptor modulator. S1P1 is found mainly on lymphocytes, so we know that targeting S1P1 reduces or prevents the egress of pathogenic lymphocytes into the central nervous system, thereby dealing to some extent with the focal damage. S1P5 is very interesting. S1P5 is found mainly inside the central nervous system. It is found mainly on oligodendrocytes. These are cells that are responsible for remyelination and repair. Preclinical studies in animals show that the interaction between siponimod and S1P5 inside the central nervous system may be beneficial in terms of repair mechanism in this progressive patient population. This is, of course, based on animal models, and it's, of course, extremely important to say that siponimod very easily gets into the central nervous system. It has almost no barriers getting there. So we have, I would say, good reasons to believe that there may be a dual mechanism of action, and this was actually the hypothesis that led us to embark into this huge, I would say, the largest ever conducted 1,600 patient study in secondary progressive MS.
So there might actually be a repair mechanism going on in this treatment as well.
Theoretically, yes. We haven't been able to show that in patients. There are animal studies that indicate that this may be the truth, but eventually going back to patients, 21% reduction in confirmed disability progression 3 months, 3 months, or 26% reduction at 6 months is something that we have never seen before with other anti-inflammatory drugs.
In early research, siponimod seemed to be effective in relapsing patients as well. Are you planning, as a company, to pursue approval for both relapsing and secondary progressive?
So that's a great question. We need to clarify that indeed in the BOLD study, the phase 2 study, we tested siponimod in relapsing remitting MS patients, but the purpose of this study was to have a proof of concept and mainly dose range finding to test the right dose in MS patients. So the endpoint in this study was not relapses and was not disability, it was accumulation of MRI lesions, active MRI lesions, just to nail the dose, the right dose. Given the preclinical evidence that suggests repair mechanism, we went to SPMS, and the hard clinical endpoints of disease progression were tested in SPMS, so we are aiming for a label in secondary progressive MS, where the biggest unmet need is.
Might you come back at a later date and look at it, especially if the repair mechanisms are indeed found to show efficacy and show the possibility in patients?
Coming back to our MS?
Right.
Currently, there are no plans. We are focused on the huge unmet need. A little bit about the philosophy of Novartis. We are really committed to multiple sclerosis, and we believe that patients should get the right treatment at the right time, with the right level of evidence. The right level of evidence for this mechanism of action currently, and the data that we have suggest that it will be much efficacious in the this advanced relapsing population, which converts to SPMS, and this is where we would like to target, also in terms of access.
Thank you. It's a well-met need. Can I ask, is there anything else you would like to discuss, highlight, or anything on other data you might be presenting, that Novartis might be presenting, that you would like to discuss?
Yeah, there is. I'll tell you a little bit about the philosophy of Novartis. Novartis has, in terms of research, we are innovating not just in new mechanism of action and new targets, which we love because we would like to bring safe and efficacious therapies to patients. We are also innovating in, I would say, pure science, but relevant science. First of all, science that will allow us to continue innovating in terms of drugs, but also science that will actually provide, which will actually be able to inform medical practice. One of the things that we're very excited about is neurofilaments. Neurofilaments are proteins. They are thread-like proteins that are found only inside neurons. They actually function as the structural support to neurons, and they're not supposed to get out of neurons if the neuron is intact. However, when a neuron or an axon is damaged, these neurofilaments spill out and go extracellularly. So until a few years ago, you couldn't measure neurofilaments in the cerebrospinal fluid, i.e. you had to perform a lumbar tap, which was okay for occasional research as a biomarker, but in terms of convenience, it was far from being convenient. Novartis was a great supporter of making neurofilaments measured in the blood. And this is, I would call it maybe cautiously, but not less cautious, I would call it revolutionary. It goes like this. We went backwards to the Freedoms trial, to the placebo-controlled, one of the pivotal studies with fingolimod, with Gilenya. It was a placebo-controlled trial, 2 years trial. And apart from the fact that we showed marked effect of fingolimod, of Gilenya, on neurofilaments, reduction of neurofilaments suggested that it reduces axonal damage. We also looked forward and tried to understand how the neurofilaments behave, whether they can predict disease progression, whether they are correlated with long-term confirmed disability progression, with longer-term brain volume loss, with longer-term accumulation of lesions. And there is a high degree of correlation between baseline neurofilaments and what happens to the patient on the long term. In parallel, neurofilament levels at month 6 correlated very well with what happens at month 24. Why am I focusing on month 6? Because most of the proof of concept studies, like the BOLD study or other studies, are being performed with a duration of 6 months with MRI. So imagine that we could have blood neurofilament levels as biomarker instead of MRI machine, a test that takes between 30 minutes to 1 hour. Blood test, and that's it. Instead of MRI, much smaller sample sizes and much faster drugs to patients. So this is extremely exciting for us.
Excellent. And do you expect that it will actually replace MRI at some point, that this will be such a solid marker that...
Currently it's under exploration.
Right.
We need to be sure that we have the right normal values. It's a long shot, but it's a very reasonable shot. You know, Novartis, starting 4 years ago, we were the pioneers in the introduction of brain volume loss, for example. We brought this concept into the concept of no evidence of disease activity, and we are planning additional activities of bringing the neurofilaments concept as a complementary to MRI. And who knows, maybe at the end of the day it will be a part of clinical practice.
So thank you very, very much.
Thank you so much.
Absolutely.